Module number 29. Photosynthesis 1. Absorbing and capturing light energy. Specialized plant pigments capture light for photosynthesis. Eucalyptus stylophermilia, one of the many species of gum trees native to Australia, leaves must capture sunlight and CO2 while minimizing water loss. Topics covered in this module. Plant pigments and light. Harvesting light energy. Major objectives of this module. Explain how photon/wave nature of light as it relates to photosynthesis. Interpret an absorption spectrum and an action spectrum. Identify 3 photosynthetic pigments. Describe the function of a photosystem. Describe the locations of photosynthetic pigments and how they are involved in the process of photosynthesis. Plant pigments and light. The sun provides the energy that most organisms require to function. Animals cannot use this energy directly, but instead consume the energy that is fixed by photosynthesizing organisms, which form a base for the next food web or most ecosystems. Chances are that if you look out your window, you will see something green. Green is the color of chlorophyll, from Greek chloros and phylon, leaf, a molecule that is vital for photosynthesis. When plants and algae and some protists and bacteria perform photosynthesis, they capture the sun's energy and convert it to biochemical energy. Oxygen production, i.e. oxygenic photosynthesis, uses light energy to split water molecules. The biochemical energy generated from light is used to convert CO2 into sugars via the Calvin cycle. The oxygen from water is released into the air as a byproduct. All of this occurs as an extraordinarily fast time scale. The capture of light energy is the first part of photosynthesis. The conversion of energy into biochemical energy and ultimately to sugars will be converted to the following molecules. Here we examine the cellular machinery that takes the biochemical conversion possible. The main cast of characters is small: the sun, chlorophyll, and other light-capturing molecules. Our star actor is chlorophyll. Chlorophyll is a pigment. Pigments are molecules that selectively absorb light energy in plants and other photosynthetic eukaryotes. Photosynthetic pigments are found exclusively in membrane-bound organelles called chloroplasts. Each one is about the size of a bacterium. Chloroplasts contain a continuous, highly folded internal membrane called the thylakoid, in which chlorophyll is bound. To understand how the pigments inside these tiny chloroplasts absorb light for photosynthesis, it is first necessary to understand the nature of light. Different plant pigments absorb different wavelengths of light. The sun produces a broad range of electromagnetic energy that radiates through space in waves. The electromagnetic spectrum is the full range of electromagnetic wave energy. The distance between 2 successive crests, the top of the wave, or troughs, the bottom of the wave, is a wavelength. The electromagnetic spectrum ranges from short wavelength gamma rays, which are less than a nanometer, to long wavelengths called radio waves, which are over a kilometer. The part our eyes can perceive in the plants occupies a very narrow part of the spectrum called the visible light, which ranges from 400 nanometers violet and to 700 nanometers red. Figure 1 is the visible spectrum. Light spectrum: a visible light which is used during photosynthesis, which ranges from 400 nanometers violet to 700 nanometers red. Given the enormous range of radiant energy, it might seem coincidental that we see the same electromagnetic spectra that plants use for photosynthesis, but it is not. The sun emits energy across a broad range of wavelengths, most of which are absorbed by particles in the Earth's atmosphere. Most solar energy that actually reaches Earth is the visible wavelength range. While some organisms can use light energy outside of the visible range, bees for instance can see ultraviolet light, most organisms on Earth evolved in the response to the light that is available, the visible spectrum. Light travels in waves, but also has the characteristics of particles. Light particles are packets of energy called photons. Photons of light are shorter wavelength and end the spectrum, and at the shorter wavelength end of the spectrum have great amounts of energy than photons at longer wavelengths. Chlorophyll absorbs photons in only some wavelengths of the visible spectrum, at the lower end and the upper end, but not in the middle. Plants possess other pigments that absorb photons in different wavelengths. Pigments state the color red of the light they reflect, not the color in the light they absorb. Knowing this, why are plants green? Plants are green because the chlorophyll reflects green light. Growing most plants under green light would likely kill their plants. Other pigments absorb some green and yellow light, but most light is the part of the spectrum is not used for photosynthesis. Plants contain two major classes of photosynthetic pigments, chlorophyll and the catharoids. There are several varieties of chlorophyll. Land plants and green algae contain only chlorophyll A and chlorophyll B. Bacteria and the cyanobacteria contain different forms of chlorophyll, one of which was discovered in 2010. This chlorophyll, found in the cyanobacterium in Australia, can actually absorb light in the infrared part of the spectrum. Catharoids, photosynthesis, found in red algae and cyanobacteria, and other pigments absorb light energy in the wavelength range inaccessible to chlorophyll. They pass this energy along to chlorophyll for use in photosynthesis. By far the most abundant chlorophyll in green plants is chlorophyll A. To us, chlorophyll A appears green in color because it absorbs best in the purple and red wavelengths. Chlorophyll B appears yellow-green because it absorbs best in blue and orange wavelengths. The molecular structure of these two chlorophylls are identical save for the substitution of the aldehyde in chlorophyll B for methyl group in chlorophyll A. Figure 2. This slight change in a functional group is sufficient to alter the molecule's absorption properties. Chlorophyll molecules have a magnesium atom contained in a structure called porphyrin ring. Interestingly, hemoglobin, the oxygen-carrying molecule in red blood cells, has porphyrin structure nearly identical to that of chlorophyll. So vitamin B12, the major differences are that chlorophyll is built around a magnesium ion and hemoglobin is built around iron, and vitamin B12 is built around cobalt. The similarity along these structures suggests an evolutionary relationship. Figure 2 is the chlorophyll structure. Chlorophyll A and B are identical except for the occurrence of a methyl CH3 group in the chlorophyll and chlorophyll A and an aldehyde group CHO in the chlorophyll B. The molecule's ring shape facilitates electron movement around its edges. The hydrophobic tail at the other ends acts like an anchor and holds the molecule in place. Each type of pigment in a plant absorbs different light at different wavelengths. Chlorophyll A is involved directly in the conversion of light energy into chemical energy. Chlorophyll B is considered an accessory pigment because it does not play a central role in the energy conversion process, but it plays a central role in light harvesting. The catharoids are also considered accessory pigments. Catharoid pigments in plants absorb the best in the purple and blue wavelengths and so appear orange or yellow. Figure 3 shows the wavelength absorbed by the chlorophylls and catharoids. Terrestrial plants contain more chlorophyll pigment molecules than catharoids, so the catharoids are hard to see. But in the fall, the temperate regions, when many deciduous plants degrade their pigments, the chlorophylls disappear first, leaving the orange and yellow catharoids vividly apparent. They are also apparent in many types of catharoid-rich marine algae, such as red algae, whose environments tend to be higher in green light. This is because water tends to scatter blue light and absorb red and orange light. Therefore, catharoids are well suited to absorb the green light left behind. Remember, chlorophyll A absorbs in the violet, blue, and red range of light. A spectrophotometer measures the pigment's absorption spectrum. The graph in Figure 3 is called an absorption spectrum. An absorption spectrum is a graph of light absorbance versus wavelength. It shows what percentage of light a substance absorbs relative to its wavelength. Scientists construct absorption spectra using spectrophotometers. A spectrophotometer is an instrument designed to measure the amount of light absorbed by a particular material. How does a spectrophotometer work? A prism splits light into separate wavelengths so that it bends or refracts into different colors. Only one color with a specific wavelength passes through the pigment sample into the correlate at the time. A photodetector, on the other side, measures how much time the pigment absorbs or transmits. It adjusts the wavelength of light to be set through the sample. The operator of the spectrophotometer moves the slit, which serves to infract the incoming light, or adjust the angle of the prism. Figure 3. A spectrophotometer. A spectrophotometer shines incident light of a set wavelength into a sample. The amount of light that is transmitted through the sample is recorded electronically on a photodetector and is used to determine the amount of light and the sample absorbed. Rotating the prism changes the wavelength that shines on the sample. How can we determine and interpret a pigment's action spectrum. A spectrophotometer will indicate how efficiently individual plant pigments absorb different wavelengths of light, but spectrophotometer data cannot indicate the overall efficiency of photosynthesis. An action spectrum measures the effect of photosynthesis across the visible light spectrum. Efficiency of photosynthesis is measured by comparing the oxygen output or carbon dioxide input of chloroplasts when they are exposed to various wavelengths of visible light. Therefore, the action spectrum will indicate under which wavelength photosynthesis occurs at the fastest or slowest rate. An action spectrum is plotted as a photosynthetic rate versus wavelength. Figure 5, the action spectrum. The action spectrum shown here is derived from a measurement of oxygen production in a plant leaf. How does it correspond with the absorption spectrum for this plant leaf? Why does the action spectrum in Figure 5 look so similar to the absorption spectra in 3? The graph of the absorption spectra of the chlorophyll A, chlorophyll B, and the cataloids look like a graph of the action spectra of a leaf because the chlorophylls and the cataloids are the only photosynthetic pigments that are present in the leaf. Because the harvesting of light by plant pigments is central to the light reactions of photosynthesis, the rate of photosynthesis is highest at the same wavelengths that correspond to the highest light absorption by photosynthetic plant pigments. Conversely, the rate of photosynthesis is lowest at the wavelengths that correspond with the lowest amount of light absorbed by these pigments. Suppose a bacterium has a chlorophyll A and its only photosynthetic pigment. What would the action spectrum of its bacterium look like? It would look similar to the absorption spectrum from chlorophyll A. Photosynthesis can only occur at the wavelengths at which photosynthetic pigments can absorb light. The rate of photosynthesis. Photosynthesis occurs at different rates depending on the quality and quantity of the light a plant receives, as well as the amount of water, carbon dioxide, and other limiting nutrients available for the plant. Differences in light quality reflect differences in spectral content. Plants photosynthesize faster and more efficiently when they are exposed to the wavelengths in which their pigments absorb more strongly. How does one determine the rate of photosynthesis? Scientists measure the rate of photosynthesis using a variety of methods, but most are based on the simple fact that photosynthesis uses carbon dioxide and releases oxygen, two measurable quantities. Therefore, an action spectrum could be used to by exposing a plant leaf to sunlight and measuring the amount of CO2 consumed or O2 produced under all wavelengths of light. Figure 6. Engelmann's crude action spectrum. T.W. Engelmann exposed an algal filament to light shine through a prism. The location of oxygen-seeking bacteria revealed the wavelengths under which photosynthesis occurs at the highest rate. Modern atomic action spectra are much more precise. In 1883, the German botanist Theodor Wilhelm Engelmann recorded a crude action spectrum or filament of algae. His experiment was elegantly simple. He introduced a population of aerobic, oxygen-seeking bacteria into a slide with the algal filament. Then he shined light from a microscope lamp through the prism into the algal filament, observing where the bacteria concentrated themselves. Oxygen-seeking bacteria and wavelength. The microscope light has not yet been turned on. Click on the locations of the algal filament where you expect to see the greatest concentration of oxygen-seeking bacteria. When you are finished, click the light switch. Did the bacteria concentrate themselves when you predicted? The bacteria clustered around parts of the algae that was producing the most oxygen. As we have seen from the observation action spectra, the sections of algae and the blue, violet, and red light with the undergoing photosynthesis at the fastest rate. Therefore, oxygen production is the highest in these areas, causing the oxygen-seeking bacteria to concentrate in these areas. Harvesting light energy. The accessory pigments such as chlorophyll B are carotenoids. Do not transform light energy directly into chemical energy. So what do these pigments do? They are simply light harvesters. They absorb light and pass the energy through other pigments. Because accessory pigments absorb more light at different wavelengths than chlorophyll A, a broader range of light can be used for photosynthesis. Before we discuss how they do this, let's step back and take a look at the big picture. Consider a leaf from a plant. A thyroid membrane of the chloroplast contains the light-harvesting machinery required for photosynthesis. Unlike the round outer membrane, the thyroid membrane is folded into stacks called grana, singular granum. When the edges of a leaf are magnified, dozens of chloroplasts that populate each mesophyll cell become visible. Mesophyll, from the Greek word meso, meaning middle, and phyll, meaning leaf. Inside the chloroplast is a continuous internal membrane called the thyroid. Some thyroid regions are stacked in layers called grana, while other parts of the membranes are unstacked called laminae. Chlorophyll are category pigments and embedded in the thyroid membranes. Each pigment is bound tightly to one or more proteins and grouped into large aggregates called photosystems. Between 200 and 300 pigment protein complexes from a single photosystem. There are hundreds of photosystems in a chloroplast. Each contains a light harvesting complex and a reaction center complex. What do these complexes do? Pigments transfer energy to other pigments in the light harvesting center. The light harvesting complex is the photosystem's light collector. It is sometimes called an antenna. It works somewhat like a satellite dish, collecting energy and concentrating it in a receiver. The receiver in this analogy of the reaction center complex. The reaction center complex is the actual site where the energy conversion reactions of photosynthesis take place. The pigment in the reaction center complex is a special form of chlorophyll A. How do pigments in the light harvesting complex funnel energy to the reaction center? The pigments and proteins in the light harvesting complex are highly organized. The proteins provide scaffolding, positioning of the pigments to optimize light absorption. When photons are just right wavelengths strike a pigment molecule. One of the molecules' electron becomes excited as it absorbs the photon's energy. The incoming light energy boosts the electrons into an outer orbital shell. The electron is unstable here. To return to this stable resting area, it must rid of itself of the energy. It passes to another neighboring pigment and then falls back until another photon strikes it and the process starts all over again. Meanwhile, the pigment that received the energy passes it, in turn, to another pigment. The energy mitigates until it reaches the reaction center. When most non-pigment molecules absorb light and become excited, they release their energy as heat and fluorescent light. Even a pigment isolation from the photosynthesis complex releases photon energy as heat, but because of the photosystem's unique organization, energy transfer to the light harvesting complex happens so fast on the order of femtoseconds, 10 to the negative 15 seconds, that hardly any energy is dissipated as heat or light. Note that the pigment molecules do not transfer electrons. They transfer only energy via a mechanism called resonance energy transfer. Energy passes from shorter wavelengths, high energy, to long wavelengths, low energy. Therefore, pigments in the outside of the light harvesting complex absorb light at shorter wavelengths than those on the inside closer to the reaction center. This energy trapping system is extraordinarily efficient. Scientists estimate that 95 to 99% of captured light funnels to the reaction center. Test yourself. Chlorophyll A is the photosystem's reaction center. Would this chlorophyll A absorb light energy at a longer or shorter wavelengths? Because light to the photosystem antenna complex funnels the pigments that absorb short wavelengths, 450 nanometers of light energy, light to pigments that absorb long wavelengths, 700 nanometers low energy light. The pigment at the very center should absorb light close to 700 nanometers of light. Why are there so many pigments in light harvesting complex? Each pigment absorbs light only at a particular wavelength range. Having different kinds of pigments ensures that the plant gathers light of as many wavelengths as possible. The size of the light harvesting complex differs in plants depending on the plant's environmental shape. Shade-adapted plants tend to have larger light harvested complexes than sun-adapted plants do. Carotenoid pigments protect plants from too much light. Some plant pigments in light harvesting centers do more than simply transfer energy. They also serve to protect the photosystems when there is too much light for the reaction centers to handle. It might seem odd that a plant would receive too much light, but this can be a serious problem for some plants. Imagine, for example, a small plant growing underneath a tree that has been recently cut down. Exposing the small plant to more sunlight, the continual bombardment of energy can overwhelm the reaction centers and cause damage to the photosynthetic machinery. Plants have devised many physical ways to reduce the impact of high light heat levels. The levels of some plants turn to edge or fold up to avoid direct light. Other leaves are covered in hairs called persibs that reflect light to make it on the leaf to appear white. In addition, chloroplasts can mitigate for more shaded parts of the cell. However, each of these responses is relatively slow. Plants also exhibit very fast psychological responses that involve cannabinoids. A major role of the cannabinoids is to protect plants from too much light. When a plant receives excessive light, cannabinoids continue to absorb energy, but instead of funneling to chlorophyll, they dissipate it harmlessly as heat. The dissipation of excess energy as heat is called quenching, because the process quenches or suppresses the excitation state of the light-harvesting chlorophylls. One well-studied quenching response in plants is called xenophil cycle. Xenophils are types of cannabinoids that come in several forms, one of which is violoxanine. Other normal conditions, violoxanine functions as an antenna pigment, but when light becomes excessive, violoxanine is enzymatically covered in zeaxanthin. Zeaxanthin, normally absent in the thyroid membrane, suppresses energy transfer by dissipating energy as heat. When light conditions return to normal, zeaxanthin is enzymatically converted back to violoxanine. Carotenoids serve a protective function not only in plants. They also play an essential role in protecting skin and the retina of eyes in animals, including humans, from the damaging effect of light. How do we obtain carotenoids? They come from eating plants. What are carotenoids? What are carotenoids found and what do they do? Where are they found and what do they do? Carotenoids are accessory pigments in photosynthesis. In plants, they absorb light in the blue wavelengths of light, more energy than either chlorophyll or chlorophyll B. As a result, having carotenoids expands the range of amount of visible light available to plants for photosynthesis. Exceptions to this rule. There are a few non-photosynthetic plants. For example, the Indian pipe Manola uifolia does not perform photosynthesis. It absorbs nutrients from the ground, much like a fungus. This species is the family Ericaceae, which includes plants like healers and blueberries. However, M. uniforma does not produce photosynthetic pigments, which are necessary for photosynthesis to occur and give plants their green, yellow, orange, or brown colors. This plant, the Indian pipe Monotropha unifolia, this plant lacks photosynthetic pigments so that it does not appear to have any color or in its tissues. However, it is able to absorb energy and nutrients from the ground. Summary. Photosynthetic organisms such as plants use visible light energy to drive the CO2 fixation that ultimately powers most ecosystems on Earth. The capture of sunlight is the only first steps in photosynthesis. The actual chemical conversion of light energy into biochemical energy and sugars begins with the transfer of energy to chlorophyll and chlorophyll A in the reaction center. Summary. Explain the photon wave nature of the light as it relates to photosynthesis. Light has the properties of both a particle and a wave. Light travels in waves, but also has the characteristics of particles. Light particles are packets of energy called photons. Photons of light at the shorter wavelength end of the spectrum have greater amounts of energy than photons at longer wavelengths. Chlorophyll absorbs photons in only some wavelengths in the visible spectrum, at the lower end of the upper end, but not in the middle. Plants possess other pigments that absorb photons in different wavelengths. Objective. Interpret an absorption spectrum and an action spectrum. An absorption spectrum is a graph of light absorbance versus wavelength. It shows what percentage of a light substance absorbs relative to the wavelength. An action spectrum measures the efficiency of photosynthesis across the visible light spectrum. Efficiency of photosynthesis is measured by comparing the oxygen output to carbon dioxide input of chloroplasts when they are exposed to the various wavelengths of visible light. Therefore, the action spectrum indicates which wavelengths of light result in the fastest or slowest rates of photosynthesis. Objective: identifies 3 photosynthetic pigments. Chlorophyll is the main photosynthetic pigment in plants. Pigment absorbs light energy at certain wavelengths and reflects the light that they don't absorb. This is why plants are green. Chlorophyll absorbs violet, red, orange, blue light but reflects green light. Chlorophyll in plants is present in 2 varieties, chlorophyll A and chlorophyll B. Plants also contain in fewer numbers cataract pigments which absorb light mostly in the purple or blue part of the spectrum. Cataract also serve to protect photosynthetic systems from too much light. Objective: describe the function of a photosystem. A photosystem contains hundreds of pigment protein complexes and is organized into 2 parts, a light harvesting complex and a reaction center complex. Pigments in the light harvesting complex gather light in front of the energy from pigment to pigment in the reaction center complex. Electrons pass from the highest energy at the periphery of the photosystem to lower energy in the middle. The chlorophyll A in the reaction center has the lowest energy. Objective: describe the locations of photosynthetic pigments and how they are involved in the process of photosynthesis. All photosynthetic pigments are embedded in the thyroid membranes of chloroplast. A plant cell contains dozens of chloroplasts, and each size of a bacterium. Accessory pigments such as chlorophyll B and cataract absorb light and pass the energy to other pigments. Because accessory pigments absorb more light at different wavelengths than chlorophyll, a broader range of light can be used for photosynthesis. A special form of chlorophyll A is located at the center reaction of complex where the energy conversion reactions of photosynthesis take place. Key terms: absorption spectrum. The graph of the percentage of light absorbed as a function of light wavelength. Action spectrum. Graph depicting the efficiency of photosynthesis of an organism across a visible light spectrum. Carotenoid pigment. An accessory pigment that absorbs light energy primarily in the purple and blue wavelengths. Chlorophyll A. One of the pigments in photosynthesis, absorbs best in red and blue wavelengths. Chlorophyll B. One of the pigments in photosynthesis, absorbs best in blue and orange wavelengths. Chloroplast. Organelle that contains chlorophyll and carries out photosynthesis. Electromagnetic spectrum. Full range of electromagnetic energy. Light harvester. A pigment that absorbs energy and passes it to other pigments. Light harvesting complex. A complex of proteins that serves as a light collector of a photosystem. Photon. A particle of light. Photosystem. A collection of pigment protein complexes organized to harvesting light energy. Pigment. A naturally occurring molecule specialized for absorbing light energy and which can be coupled to energy-producing mechanisms in photosynthetic organisms. Exist in a variety of types so that absorb light at a specific wavelength. Reaction center complex. Study the energy-converting reactions of photosynthesis. Spectrophotometer. An instrument used to measure the amount of light of a given wavelength that a sample absorbs. Thyroid membrane. Membrane within the chloroplast in which many photosynthetic pigments are embedded. Visible light. Narrow portion of the electromagnetic spectrum ranging from 400 to 700 nanometers, corresponding to the colors visible to the human eye. Wavelength. The distance from one wave crest to the next successive wave crest, or wave trout.